So you are welcome again. We expecting we experienced network problem, but I hope so. Please, I want someone to write in the group for those of you that joined this last link. So please write in the group that this last link is the one that we are going to use. So the network problem is from my side. So I think I identify the issue. So I hope the class will move quickly now. So what about the glycoproteins found in blood group O? So actually in the blood group O individual, Fatima Adam, for somebody with blood group O, it means that the N-acetyl galactosamine and the galactose are absent on the red blood cell, and that is why they are considered as blood group O individual. And in fact, that is why they are even universal donor, because that sugar is what actually caused the cross reactivity. It is those sugar that cause that cross reactivity. If you're actually going to check or diagnose somebody, or if you want to check the blood groups of a particular individual, then you use antisera. So why are you using antisera? Because the glycoproteins there is the antigen. So the antigen that found on an individual with blood group A, it means that on the surface of the protein, that is the antigen, there is an N-acetyl galactosamine. And then for the individual with blood group B, it means that there is for individual with blood group B, there is galactose. For somebody with blood group AB, it means that they have both N-acetyl galactosamine and galactose. And then for someone that actually have blood group O, it means that both N-acetyl galactosamine and galactose are absent. So this is actually the description that we are going to explain in question 1b. We are going to explain it based on the concept. Okay, so please, are you hearing me? Can you see the slide that I'm sharing? And also, can you also hear me at the same time? Okay. So I hope you get the answer for question B now. Do you get the answer for question B? Okay. So now we are moving to question 1C. Question 1C says sequentially list and describe the 6 classes of an enzymes. So this is 6 marks. What are you saying about no sir, no sir? What is the problem? This, what is the problem? Okay, so okay, someone says 1B is not clear. So the 1B says the blood or the ABO blood group system is related to some glycoproteins discussed. So I will say it again. So generally, ladies and gentlemen, we know that humans have different blood group system. We have individual with blood group A, individual with blood group B, with blood group AB, and uh, an individual with blood group AB. So now the question is, why these blood groups vary across individual? So ladies and gentlemen, what make the blood group system differ from one another is because of the sugars that are found on the red blood cells. It is because of the sugars that are found on the red blood cells. So an individual with N-acetylgalactosamine on the surface of the red blood cells, that individual is said to have a blood group A. While an individual with blood group B, they have galactose on the surface of their red blood cells. While the individual with blood group AB, it means that they have both N-acetylgalactosamine and galactose. That is why they have blood group AB. The A, the galactose, the N-acetylgalactosamine is what make the individual to have both blood group AB. While in the case of blood group O, because there is number one, there is no there is no N-acetylgalactosamine and no galactose. That is why they have blood group O. So is that clear now? Is that clear now? A, for an individual with blood group A, they have N-acetyl galactosamine. For an individual with blood group B, they have galactose. For an individual with blood group O, they neither have galactose, neither N-acetyl galactosamine. So this is actually the answer to question 1B. So we are now moving to question 1.6. Sequentially list and describe the 6 classes of enzymes. The 6 classes of enzymes. Okay, Diago, thank you. So now, how do we name the enzymes? So ladies and gentlemen, check my comment section. I'm going to write one simple formula. Look at it. I'm writing it ortholyl. Can you see my ortholyl? So if you ask to write the name of the enzymes, so you follow it ortholyl. I'm coming. It's not ortholyl. Yes, ortholyl. So this is the correct spelling. Ortholyl. Check my comment. Ortholyl. Othlil. So the first O, you can jot it down. The first O stand for oxidoreductase. Oxidoreductase. That is the first one. Oxidoreductase. Oxidoreductase. So I wrote it on the comment. Check it. Oxidoreductase. That is the first enzymes. So let me finish with listing the enzymes, and then after listing the enzymes, then I will describe each and every one of them. So ortholyl, the first one stand for oxidoreductase, and then T stand for transferase. Transferase. That is the O, so that is the T, and then the H is hydrolase. Hydrolase. That is the third one. And then the L is lyase. It's lyase. That is the first L. And then the I is isomerase. Is isomerase, and then the last one is ligase. Ligase. So this is the sequential listing of an enzyme. Ortholyl, first O stand for oxidoreductase, the T transferase, hydrolase, lyase, isomerase, and ligase. These are the 6 classes of enzymes. So I'm now going to describe each and every one of them. The oxidoreductase they catalyze redox reaction. They catalyze both oxidation and reduction reaction. And then transferase, It's a group of enzymes that involve in a transfer of group. What they are doing is to transfer a group, like for example, they can transfer phosphate, they can transfer sugar. It's just a very simple, you take one group of compound from here and then you take it from here, you transfer it. So that is the transferase enzymes. And then the next one is hydrolase. The hydrolase catalyze hydrolysis. Hydrolysis. What is hydrolysis? Hydrolysis means breaking down of compound with addition of water. So you add water molecules and then you break it. Hydro means water. The lysis from the word hydrolysis, it means you use the enzymes to break down a molecules with addition of water molecule. So that is hydrolysis. Lyase is also a breaking down. Isomerase is a group of enzymes that catalyze isomerization, while ligase are the group of enzymes that join. Let's say you have one molecule here, then you have another molecules here. So the ligase, they are group of enzymes that join two molecules together. They join two molecules together. So ladies and gentlemen, is 1C clear now? I'm waiting for your response. Is 1C clear? Can you hear me, please? Okay, thank you. So 1C is clear. So we are now moving isomerase. Isomerase, okay, Sandra, isomerase are enzymes or a group of enzymes that catalyze isomerization reaction. Isomerization. Yes, sir, please come again on transferase. Transferase are enzyme that transfer a group of molecules or a group of compound from one compound to another. It's just a transfer. Yes, sir. Can you repeat oxidoreductase? Yes, they are enzymes that catalyze redox reaction. When we said redox reaction, it means reduction and oxidation reaction. So oxidoreductase are the group of enzymes that catalyze uh, redox reaction. Ligase, they join two molecules together. Ligase, you have molecules A, you have molecules B. So a ligase, a group of enzymes that join two molecules together. Lyase, it breaks down. When we said lyase, it means it breaks down a molecules. Like for example, it's like lyase is an opposite. Lyase is an opposite of ligase. Instead of joining it, break down. Let's say you have a molecules like this, then you break it, while ligase joined together. So please, ladies and gentlemen, do you understand these classes of enzymes with their brief description? I hope you can still see the PDF. Can you clearly see the questions that I shared? I'm waiting for your response, ladies and gentlemen. Okay. So now we are moving to question D. Question 1D. Question 1D. Question 1D says, what is the relevance of B vitamins and their derivatives to catalytic reactions in our body systems? So generally, ladies and gentlemen, let me say this. Uh, if you can remember, we have talked about uh, enzymes. We said enzymes are organic catalysts that speed the rate of the reaction. If the reaction is going to take, let's say, 10 years with an enzymes, it can take just few seconds. In fact, some reaction take less than seconds. But remember, I told you that we have pro enzymes, we have hollow enzymes. What is pro enzymes? Pro enzyme, pro enzymes, it means they are inactive enzymes, while hollow enzymes are active enzymes. So some of these enzymes need cofactors or coenzymes for them to work. And in the absence of these coenzymes or cofactors, the enzymes will remain inactive. And in the absence of these coenzymes or cofactors, the enzymes will remain inactive, derivatives to the catalytic reactions in our body systems. So generally, most of the coenzymes, ladies and gentlemen, get my point. Coenzymes, there are molecules that are needed by the enzymes for them to become active. So we have vitamins. So the vitamins are the coenzymes that are used. The coenzymes are vitamins that are required by the enzymes for them to work. If there is no coenzymes, then they are not going to work. So most of the coenzymes, they belong to vitamin V, especially vitamin V3, vitamin V1, and vitamin B12. So we have number one, we have these vitamins. So these vitamins, they are coenzymes and they are required. Okay, are you saying that the PDF is not showing? So let me share it again. So vitamin V, we have niacin, we have flavin, we have riboflavin. So you see, niacin, flavin, riboflavin, they are the group B vitamins that are strongly associated with the activity or with the functions or the catalytic activity of the enzymes. So in the absence of these vitamins, that is coenzymes, the enzymes become inactive. So ladies and gentlemen, do you understand this? So vitamin B12s, they are very essential, they are very important by the enzymes for them to work. So that is the answer to this. So ladies and gentlemen, can we go to question 2A? Can we go to question 2A? Okay, what I said, vitamin B12, they are a group of molecules that are actually considered, or they are called coenzymes. And these coenzymes, they are molecules that are required by the enzymes for them to work. So in the absence of vitamin B12s, by the absence of B vitamins, some of the enzymes are going to remain inactive, so they are not going to carry out enzymatic activity, they are not going to catalyze any reactions. So vitamin B12s, they are very essential and they are very important in catalyzing most of the biological reactions in the body. So that is what I said. And under these vitamins, the ones that are mostly used by the enzymes, we have niacin, we have flavin, and we have riboflavin. For those of you that are yet to join the YouTube community, try to join. I'm saying this so that at least immediately after your exams, we are going to focus on general metabolism, general microbiology, and general physiology. So that is why I always encourage you to join the YouTube community. So then the next thing that we are going to look at is the highlight the relevance of human genome projects in medical biochemistry. Yes, the vitamins are the examples of coenzymes to carry. Yes, to carry. The vitamins that I mentioned are the examples of the coenzymes. Okay, Ijafa, in essence, it is a coenzymes or a molecules needed to activate the enzymes to work. Yet it is the coenzymes are the molecules. So the coenzymes they are needed to activate the enzymes for them to work. Ijafa, yeah. So now, and I mentioned the members of the vitamin V that are the coenzymes that are mostly used to activate the enzyme. I said we have niacin, we have flavin, we have riboflavin. So now the next question is to highlight the relevance of Human Genome Project to medical biochemistry. So actually, as a result of the Human Genome Project is a project that started in 1990 and ended at 2003. So it takes scientists 13 years to sequence the complete human genome. And in for the human genome, there are about 3 billion base pairs. And this project is one of the largest projects that conducted by the scientists. In fact, as a result of this human genome project, scientists was able to actually yeah, the narration of medical and life sciences has changed because since the human genome project was conducted, scientists was able to understand the knowledge of so many diseases. Like for example, before we don't know anything concerning sickle cell. We know that yes, there is a sickler, but we don't know the mechanisms and the etiology of the sickler till when the human genome is completely sequenced. So now, what are the relevance of human genome project? So after the human genome project is completed, it helps scientists in the treatment of diseases. That is one. So this is I am highlighting the relevance of human genome project in medical biochemistry. So it helps in disease diagnosis because since then, if you have a particular patient and maybe you don't know whether the person is actually having a genetic disease or the disease is actually a genetic, so what you are going to do, you can just take you can isolate the DNA from that particular patient. You can sequence the complete genome and you can compare it with the normal person. And from there, you will be able to know where exactly the problem is. So that is why using Human Genome Project, scientists was able to diagnose so many diseases. Like for example, let me just give you this example. Remember we talked about DNA. So a DNA. Okay, Atika Ilyasu, you are welcome. Thank you for joining my YouTube channel. So what I am saying now is that in essence, before how scientists was able to know the etiology of sickle cell anemia. You see, it's just a change of adenine as a nucleotide. It's just a change of adenine as a nucleotide with thymine. And as a result of that change, it code for amino acid valine. So, ladies and gentlemen, it's just one nucleotide, one change in nucleotide that changed the history and the narration of the sickle cell disease. So that is how scientists with the knowledge of human genome project they were able to diagnose disease. So the knowledge of human genome scientists was able to have in the drug development, disease treatment, as well as disease diagnosis. So these are the relevance of these are the relevance of human genome projects to medical biochemistry. So then we're moving to B. So I think you are now clear. What are you saying that you said that my face is okay? What do you mean my face is okay? Are you saying that you are not seeing the slides? So we are now moving to question 2B. Question 2B says briefly describe the functions of the three of the following organelles. Okay, the year of the project it started in 1990 and it ended at 2003. So it takes scientists 13 years to sequence the whole human genome. When we said human genome is DNA, yes, 1990 to 2000 and no, 2000, not 2013, 2003, 2003, not 13. So briefly describe the functions of the three organelles of the following organelles. We have smooth endoplasmic reticulum, lysosome, ribosome, and nucleus. So let me just actually explain them. One, smooth endoplasmic reticulum is used or is a site where drugs are synthesized. Sorry, where drugs are metabolized. So all the drugs that we are taking, all the drugs that we are taking, we metabolize it or we process it at the smooth endoplasmic reticulum. And then two, lysosomes. What is the importance of lysosomes? Lysosomes is important in the body in order to protect you against any foreign organism. Let's say you have Salmonella typhi get into your body, maybe also trying to penetrate into your cell. And luckily enough, maybe it penetrates into the cell and then uh, what will actually take care of the bacteria in the cell is these lysosomes. So the lysosomes will engulf the bacteria, and in the lysosomes, there is an enzyme called lysozyme. And these lysozymes, after chopping the bacteria, it will break it, it will degrade it, and at the end, the bacteria will be completely neutralized. And then the next one, three, is ribosomes. So a ribosome is a site of the cell or is the organelles of the cells where protein synthesis takes place. And then the nucleus, which is number four, it is where the activities of the cell or is where all the activities of the cells are controlled. So all the activities of the cells are controlled by the nucleus. So I think, ladies and gentlemen, you are clear with this. So let's move to another question. So we are done with question B. Then we are now moving to question C, 1C. Sorry, question 2C. Question 2C say that state the type of transport. I hope you can still see the slides. Question 2C says state the type of transport system involved in each of the following. No, I don't have problem of network here. Maybe it is you that have network issue. So you see, states the type of transport system involved in each of the followings. Roman Pugo 1, sodium glucose transporter. So you know that if you check the types of the transport system across the cell membrane, you know that we have made mention the types of the transport system in which like in the case of sodium glucose transporter. So in the case of sodium glucose transporter, is a type of transport system in which both sodium and glucose get into the cell simultaneously. So which types of cell is that? Sorry, which types of transport system is that? Who can tell me when you have two molecules moving simultaneously in the same direction, which type of transport system is that? Please, I'm waiting for your answer. When you have two molecules like sodium, glucose, when they are transporting into the excellent. Kaulat Emanuela, the answer is sympot. So this Roman figure 1, the answer is sympot. Then Roman figure 2, sodium potassium transporter, what is the answer? In the case of sodium potassium transporter, what is the answer? Sodium potassium transporter, what is the answer? No, it's not unipot, it's antipot. Excellent, it's antipot. So sodium and potassium are transporting. Let's solar na one charger. So when you have sodium potassium transporter is a type of the transport in which the sodium and potassium are moving across the cell membrane in opposite direction. So as the sodium is getting in, the potassium will be going out. Or as the sodium is going in, then the potassium will be going out. So that is what actually happened in the case of this transport. So as you said, the answer is antipodes. The answer to this is antipodes. Okay. Then the next one is sodium ion potassium ATPase. Sodium ion potassium ion ATPase, which is Roman figure 3. Please, what is the answer to this one? That is Roman figure 3. Who can answer it? Okay, the answer, active transport. Yes, I can agree, is active transport. Is active transport because it's a type of transport that require the energy, that is ATP, to transport molecules. But which types of active transport? Because active transport is of two types. We have primary active transport and we have secondary active transport. So which of the active transport? Is it the primary or the secondary active transport? Excellent, Sandra. The answer is primary active transport. Abdul kai shiru mana. So the answer is primary active transport. Then Roman figure 2. Sorry, Roman figure 2. 4. What is the answer for the types of transport that involve the release of insulin from the pancreatic cells? The release of insulin from the pancreas. What is the answer? You guys should allow the tutor explain. Stop dropping much messages. Yeah, I'm asking what is the answer for the Roman figure 4? Some are saying exocytosis, some are saying unifort. Please, which one is the correct answer? Okay, someone says facilitated diffusion. So, first, what you need to understand at this point is that the release of insulin from the pancreas is actually exocytosis. Is exocytosis. So the answer is exocytosis, because insulin is a protein and it is a very huge protein. So that is why it requires to actually because whenever we have a bulk transport, when a large molecules are moving from from inside of the cell to outside of the cell, that is why we said release of insulin from the pancreatic cells. It means that it is going out from the pancreas. So the answer is exocytosis. Okay, thank you. So now, yeah, it's not facilitated diffusion, it's not unifort. The answer is exocytosis. So now, let's move to the next question, ladies and gentlemen. So we are done with 2C. So I'm now moving to 2D. So 2D, write the important equation you use in the preparation of a buffer and define each term in the equation. So actually for the preparation of buffer, I think this one will take us time, and there is an equation called Henderson-Hasselbalch equation. So I don't have the equation here. So maybe I'm going to drop a video on this next time. So I always encourage those of you that didn't join the YouTube, please try to join. It's very, very important. It's very, very important to join so that at least you'll have access to most of these videos. So I'm going to drop videos very soon on that. And then the next one is state the mechanisms of action of each of the followings: aspirin, penicillin, disulfiram. So please, who can tell me what is the mechanisms of action of aspirin? Yes, the mechanisms of action of aspirin. Please, sir, we would like you to list out the answers, please. Okay, you want me to list the answer. So it means that if that is the case, I have to do another video, because this one I'm just only reading the questions and giving answers. Hello, do you support this? Do you want me to write the answers to this? So I think what I'm going to do, when are you going to write biochemistry? from your timetable, when are you going to write the biochemistry? But I think if you ask me to do that, why can't you just be jotting all these things? I think there is no need because the video is there. The video is there. Even if you have problem with any answer, you can still go back. You can still go back and get the answer again. So the video is on my YouTube channel and you can always have access to it. No, sodium potassium transporter is antiphot. No, Shaibu. It's not antiphot. Yeah, okay, sodium potassium transporter. Yes, I agree with you. It's antiphot. It's antiphot. Yeah, you are right. Okay, someone are saying that I should answer the last question. So please, if you want to know, if you want to get all the answers to this, what you are going to do, I advise you to go and replay the video again so that you can jot down all the answers. So now the next one is state the mechanisms of action of each of the following. Aspirin. So what is the mechanism of action as or aspirin? So actually an aspirin is a drug that used to inhibit an enzymes called cyclooxygenase. Cyclooxygenase is an enzyme that actually that induces headache, that induce headaches and pain. So with an aspirin, you can inhibit that enzyme. So once the enzyme is inhibited, then there is a likelihood the headache and the pain will be reduced. Yeah. And then what about the mechanism? So you see the mechanisms of action of aspirin is to inhibit the activity of cyclooxygenase. I have a video on the video on prostaglandin. Yes, prostaglandin is part of the course that you are doing. So prostaglandins. Okay, someone said I should come again. I said that aspirin is a drug that actually inhibit the activity of an enzyme called cyclooxygenase. So cyclooxygenase is an enzyme that actually involve in causing headache and pain in the body. Cyclooxygenase, that is the name of the enzymes. Let me write it on the comment. Cyclooxy cyclooxy cyclooxygenase. In bracket, you can call it COX. COX. That is the name of the enzyme. Cyclooxygenase. So what about penicillin? So please, if you need more answer, or if you need more explanation on this cyclooxygenase, the cyclooxygenase is an enzyme that cause headache and pain in the body. So whenever you feel pain, whenever you actually have headache, it means that the activity of these enzymes has actually goes off because it produce some prostaglandins. Some those prostaglandins, they are molecules that cause the headache. So therefore, if you want to relieve pain or headache, you need to get a molecules that can inhibit these enzymes. So ladies and gentlemen, let me just tell you this. It's not only aspirin. If you can go to the lab, identify an inhibitor that can inhibit the activity of the cyclooxygenase, it can also serve as a pain reliever. So that is how we do in biochemistry. Most of the drugs that you are seeing in chemists, most of the drugs that you are seeing, they are enzymes inhibitors. Thank you, Salma. Salma Muhammad, thank you for joining. Thank you for joining the YouTube community. So, ladies and gentlemen, is that clear? So then the next one is penicillin. Penicillin is also is an antibiotic. So what is the mechanisms of action of this penicillin? So I have to say it, ladies and gentlemen. Remember, bacteria are classified into two based on their cellular structure. We have gram-positive bacteria and we have gram-negative bacteria. So gram-negative bacteria have in their cell wall they have peptidoglycan. All the bacteria, both gram-positive bacteria and gram-negative bacteria, they have both peptidoglycan. But in the case of gram-negative bacteria, they have thick, very thick peptidoglycan, while gram-positive bacteria they have thin peptidoglycan layer. So that peptidoglycan is what actually protect the bacteria. So once you disrupt, they usually die. And the major enzymes that involve in the synthesis of peptidoglycan is called transpeptidase. So it means that if you can inhibit that enzyme transpeptidase, it means that the bacteria cannot synthesize new peptidoglycan. And if the bacteria cannot synthesize the new peptidoglycan, it will die. So penicillin is a drug that usually inhibits the activity of that transpeptidase enzymes. So once the activity of is inhibited, enzymes cannot synthesize peptidoglycan. So once the peptidoglycan is synthesized, it means that the bacteria will die. Action of penicillin. And then the next one is Roman figure 3, which is disulfiram. I don't know the mechanism of action of disulfiram. I don't know it, but I will check later. This question paper that I am answering, I didn't go it through. I didn't go through it before. So I didn't go through it before, but I will check it again. I will check it again. And when I check it, I will get back to you. So someone said I should go back again for the penicillin. I said that penicillin, let me just, their body is protected by peptidoglycan. And once the peptidoglycan layer is being destroyed, it means that the bacteria itself can also be destroyed. Enzymes that involved in the synthesis of peptidoglycan is transpeptidase enzymes. Inhibit or is designed in such a way that it will inhibit the activity of the transpeptidase. So once the activity of transpeptidase is blocked, it means that the bacteria cannot that can inhibit the activity of these transpeptidase enzymes. It means that that drug or that compound can serve as pain reliever. Sorry, it can serve as antibiotics. It can serve as antibiotics. So any compound or any molecules that can inhibit the activity of that transpeptidase enzymes, then it can actually serve as antibiotics. Salama Muhammad, thank you for joining. So this is actually for question 3A. So let's go to question 3B. Differentiate between three types of reversible enzymes inhibition. So ladies and gentlemen, uh, when we said inhibition, it means that it's when the activity of particular enzymes is blocked using a molecule. So we have different types of inhibitors. Please let me actually see if some of you knows the types of the inhibitor, the types of the inhibition. Please write it on the comment. How many types of inhibition do we have? There are three. So mention three types of reversible enzymes inhibition. Please write it on the comment section. Please.